Everyone, welcome to the 285 Exam 2 review. We're going to go through a Kahoot first. And as you're joining in, let me know if you've never played Kahoot before and you need help, but pretty much just go to that website and enter the PIN. And then you can, on your phone, I think you can do it on your computer too.
Yeah.
But the questions will show.
I am.
Yep. Yeah.
I have about only 24 questions on this one, but we're going to go over like some others after too.
Oh, sorry, I zoned out again. I'm playing this. Let me try this NVG or whatever the fuck, the shot in the dark. It's like a minute or two. I just wanted to see if it was on.
Okay. Let me know if you have any trouble joining in. We'll start in a few minutes. I wish we had like prizes to give out. I
know.
doing Kahoot in like high school and getting like, I don't know, like extra credit.
Extra credit points. I know. Maybe I can negotiate with Laura and be like, hey girl. Oh my God, this is recorded.
Give out some like biosci merch.
Yeah, literally. I don't even have the t-shirts. I don't even know where they are.
I think Shivani has the t-shirts.
Oh yeah, you're right. I think she does.
I need a new pin though, if you know where those are.
Oh, that's a good one. I don't know where the pins are. Wait a few more minutes, then we'll get started. All right, I guess we can get started. People can join in as it goes, right? I'm pretty sure they can.
I think so, yeah.
All right, let's get started.
And stop us if you have any questions or want us to go over further as we're going through this.
Mm-hmm. All right, so what would happen if the amino acid in the bad protein were mutated to an amino acid that can't be phosphorylated? Oh, I forgot to set the time to longer. So sorry.
Oh my God, what is this?
Did you see Mort?
Yeah, so
good job.
You want to explain this one, Jason?
Yeah, so bad is pretty much what so bad's job is to sequester BCL2, and I think if it's phosphorylated, then it doesn't bind BCL2 anymore. Wait, no. If it's phosphorylated, it does bind BCL2. So if it can't be phosphorylated, then it won't bind no, sorry. So bad sequesters BCL2, and BCL2's job is to kind of plug holes in the mitochondria. And so if it can't be phosphorylated, then BCL2 will be won't be allowed to plug those holes, and then the cell will just die, but it will kind of undergo cell death. Sorry, I didn't explain that very well, but any questions on that? I can try to re-explain that.
All right. Should I make the seconds longer? I feel like that was really short. I can't really think about it. I'm gonna.
Maybe, yeah.
Okay, hold on. Wait. How do I? Okay, hold on. So sorry, everybody. Just gonna quickly. I'm gonna have to redo it, but the seconds were a little too short, and I don't want you guys to be rushing through every single one. Please hold. Jason, entertain them while I do this.
Any questions or things you want us to focus on in this review? if you're coming in late, we mentioned at the beginning that this is going to be like a quick-ish Kahoot, and then we have time after to go over any questions that you have and some of Laura's practice questions that she goes over in class too. But if not, we'll just go through some of the ones that me and Rachel made in this Kahoot. And
how'd you guys feel about the first exam? How'd it go? Anyone?
The second exam will be pretty much the same style. So learn from what happened on the first exam, I guess. And it'll be very similar questions. Laura loves to, especially on this exam, Laura loves to ask mutation questions. So like you should know what happens in the pathway, but also what could happen if something goes wrong in the pathway. So that's kind of going to be the focus on this exam.
She also loves the microtubules and like the graph. I don't know if you guys remember.
Mm-hmm.
But yeah.
Yeah, last semester there was a graph question about microtubules and you had to draw out a graph for the exam. I graded that question, so I'm very familiar with that one. It was pretty much just like adding different drugs to the in vitro assay and kind of seeing what happens to the graph. So definitely know how to do that. But all right, we have to restart the Kahoot. So try to join back.
Sorry. Well, now you guys will all get the first one right. I like spice. I don't know who that is, but I like it. We're still recording, right, Jason?
Yep. All right, give it another minute and then we'll just start and people can join as we go. Put the pin in the chat in case it doesn't keep showing up. I think it does though. All right.
Sorry, that was me.
Nice.
All right, we're gonna start. Let's go. The first question is a bad question. Well, about bad, not a bad question, but about bad.
All right, maybe I can redeem myself on the explanation.
Yeah. Yay.
Nice. So BAD's job when it's not phosphorylated is to inhibit BCL2. So if it can't be phosphorylated, it can't inhibit BCL2, and which would allow the cell to undergo programmed cell death. The holes in the mitochondria couldn't be plugged, and then the cell would just die.
Wah, wah. All right. In the GPCR pathway, if G protein had a mutation for a weaker affinity for GDP, would signaling occur more or less often? So the G protein has less affinity for GDP. Okay.
Does anyone want to explain this one?
Doesn't this like go back to the whole thing with like KD or whatever? Where it's like if it's the affinity, the higher the affinity, the looser it is, the lower the affinity, the more tightly bound it is. Is that
So
KD
isn't, so it's more so like the higher the KD, the lower the affinity, the lower the KD, the higher the affinity. So it is an inverse relationship. It's not exactly like KD. So KD is just a mathematical way of explaining affinity. It's not exactly like a tell-all for what the affinity will be. It's just how scientists kind of calculate it. But in this case, so if the G protein had a weaker affinity for GDP, that means GDP doesn't diffuse in as much. So this means that the G protein is going to be more active and bound to GTP. So signaling would occur more often than normal. Does that make sense?
I have a question.
Yeah, it does. It clears it up. Thanks.
Yeah, no problem. Yeah, what's your question?
Isn't it like when it starts the pathway, isn't it bound like inactive to GDP though? Like and to initiate to start it?
Yeah, but if it has weaker affinity, it'll let go of GDP more easily. So in either case, it would let go of GDP more easily to have GTP diffuse in to start the reaction more. And then it also would not bind to GDP to end the reaction at the end either. So it kind of like if it has a weaker affinity for GDP, it's more likely to let GTP diffuse in because it like isn't as tightly bound to GDP as it normally would be.
Okay, thank you.
Yeah. Does anyone have any other questions before we move to the next one? Okay.
Nice.
All right. So if the RGS that regulates G protein activity has a loss of function mutation, would signaling occur more or less often than normal? Good job.
Nice. Anyone want to explain that?
Well, isn't the RGS like it helps like hydrolyze GTP to GDP? So if it has a loss of function, then it wouldn't really help hydrolyze it down and still be in the GDP so active state.
Yep. Exactly. So the RGS is kind of like a gap, and so it helps hydrolyze GTP to GDP. And just like you said, if it can't do that as often, it'll be more likely bound to GTP, which would increase the signaling.
Awesome. All right. Which domain allows GRAB2 to bind to the dimerized receptor?
Nice.
Good job.
Good job, everybody. The SH2 domain is a pretty common domain that this one allows your GRAB2 to bind to the RTK. Does anyone know what kind of domain is on GRAB2 that allows the next protein in the pathway to bind?
Just SO. Yep.
Exactly, SH3.
Yep. That's where SOS comes in and binds to GRAB2 with the SH3 domain. Good job, guys. All right. Haley has a streak. All right, true or false? Cells can receive multiple signals at once. Yeah, good job.
Good job, everybody. This is true. Cells can receive lots of different signals at once. And yeah, that's pretty much it.
That's why cells often have multiple different receptors, just doing a lot of different things at once.
Mm-hmm.
All right, true or false? When a signal binds to a receptor, it then always passes through the cell membrane. Yeah, good job, guys.
Nice. So it usually doesn't pass through the cell membrane, honestly. So it usually just binds to the receptor and then which then activates something downstream. Some receptor molecules do pass through the cell membrane if it's lipid soluble, but most of the time it'll just bind to a receptor outside the cell membrane, which causes something to happen inside the cell.
Awesome. Good job, guys. Yuri is a streak. Nice. All right.
I just have another question about the way that she words like her that for this in particular.
Yep.
Like I know you said like usually scenarios of like mutations or like loss of function or gain of function for like certain pathways, but like besides the graph for the microtubules, like with the cell cycle, what should we be expecting?
So so the biggest thing that for mutations is probably the signal transduction pathways, I'd say. And so any different number of mutations could happen in those pathways, which could have like different downstream effects. Usually, like for example, the RTK MAPK pathway, that's a pathway that's commonly uh, that's a cell division pathway. So it usually is something's wrong with that pathway in cancer. So you might have a mutation that leads to like overproliferation of the cell, for example. And she might ask you like, if like MAPK3 was always activated, would it cause cancer or something like that? And that would be true because it would be continually activating. Questions like that, like different things that go wrong in the pathway, would it have, what downstream effects in the pathway would it cause? And, or if you activated something like upstream, would it have a downstream effect? Something like that.
Okay.
Like an example would be like in like her previous exams, she's had like a scenario where there's like a mutation. So like one of them was like GRAB2 was missing its SH domain. So like what would happen? Like would cells differentiate? Would they not? Like what would happen like based on it missing that domain? Or she'll ask like if a cell lacks like a functional like RAS GTP or a RAS gap or something, like what would happen to like the pathway it's involved in. She did some on signal transduction, like with like certain proteins. So she talked about like BAD, JAK, GRAB2, RAS, GPCR, all that. So it's going to be a lot of like mutations, like loss of function, gain of function. She might have you draw a graph for the microtubules because she usually loves to do that. And Jason is usually the one who grades them. So make sure you like really understand microtubule formation and like the whole process of building and destroying microtubules. Like make sure you like really understand that because I can almost guarantee she'll ask you a question about that.
Okay, in particular, like with the drugs that we've learned, like Taxol.
Yeah, Taxol, colchicine. I think what is the other one? Vinblastine. Yeah, vinblastine. Yeah.
Like how would that affect the graph? Kind of is what you're trying to look for. Last semester was Taxol. It would probably be a different drug this semester.
Yeah, it's probably gonna be a different
same
one.
type of question.
Okay, and then I'm sure there's probably like a question in the Kahoot, but when it gets to it later on, and if not, like towards the end, do you mind going over the difference between crosstalk and like signal amplification? Because I kind of
have
there are a couple of questions about that, so we'll go over
that. But if like we can go over like practice too, if like the Kahoot doesn't explain it well enough, but yeah. Okay, so is tubulin an enzyme?
We were split on this one. Does anyone want to just say why it is an enzyme or why that makes sense that it is an enzyme?
It hydrolyzes GTP to GDP.
Yep. Tubulin's a GTPase, so yes, it is technically an enzyme. Anything really that ends with ACE is classified as an enzyme because it's usually speeding up a process. But yeah, tubulin technically is an enzyme. Does anyone have any questions? Kind of split on this. No, okay. All right. Another true or false. The same signaling molecule can yield different outcomes by binding to different cells. It's true or false? I struggled reading that.
Nice. Everyone got this one. That is true. So like, in what cases could a signal molecule yield a different outcome in different cells? Does anyone have any guesses? Yeah, go.
Is it because of they have different downstream effect of protein.
Yep.
Yeah.
That's like one. That's definitely one of the ways. Usually it's because there's different cell types. So like one signaling molecule might have a different effect in a neuron than like a heart cell or something, and usually that is a result of different like downstream effectors, different receptors in those different cell types. So yeah, you got it for sure.
All right. You guys are doing great. All right. RAN and RAS are a part of the same protein family of GTPases.
A lot of people were frustrated about this last semester, so I want to make sure this is cleared up for this semester. Yeah, that is very true. So they are in the same family, and that also means that GEF and GAP are like the same sort of effect in this. So like last semester, people were mad that GEF and GAP came up again on this exam because it wasn't covered explicitly since the first exam. But GEF and GAP will have the same effects on like RAS as they do RAN. So GEF is the guanine nucleotide exchange factor. So it exchanges GDP for GTP, makes it more active usually. And on the other hand, GAP is the guanine, I forget what it stands for, but it basically hydrolyzes GTP to GDP. Any questions about that?
Awesome. All right. What is the name for the process by which two parts of a receptor come together?
Nice.
Yeah, good job.
Didn't fall for my tricky.
Dimerization. Yeah, no such thing. Nice try.
Good job. Yeah, it's pretty much dimer is di just means two. So two parts of the receptor coming together is dimerization. It usually makes it active.
What is the name for the process by which a single signaling molecule can activate multiple downstream signaling targets? This is what we're talking about. It's crosstalk and amplification. Californication, that's it.
That's definitely the answer.
Mm-hmm.
I don't know. It could be a name for something.
Yeah.
Nice. Good job, guys. So does anyone want to explain signal amplification?
So when you think of signal amplification, think of it like gossiping. It's like you tell one person, they tell another, and it just gets to a lot of different people. That's kind of the same thing with this. So a single signaling molecule so that's the source of the gossip, right? And then it sends to multiple targets. It ends up with a lot of people hearing it. That's what signal amplification is. We have a question about signal crosstalk, so we can talk about the differences there. But does anyone have any questions about this? I mean, you all got it right. You answered. But
yeah, I think the gossip is like the best way to explain signal amplification.
Yeah. It gets spread around, or you can even think of it like COVID now.
Yeah, exactly.
It's a little
I was about to say that.
You could do it starts like patient 0 is the single signaling molecule, and then it gets down to like a lot of different ones. So that's like signal amplification. We used gossiping as the example when COVID was not a thing, but here we are.
Pre-COVID gossip was, but I guess COVID is definitely the better example now.
Yeah. Yeah, you're welcome. Californication is not a real thing. No one answered that, thank God, but that's not real. That doesn't exist. But yeah, if you guys don't have any questions, we can move on, but we'll talk about signal crosstalk, and I think it's actually coming up, but all right. What is the name for the process where different signaling pathways interact with each other to increase or suppress another? I ran out of characters. It wouldn't let me finish the question, so.
Oops, I didn't realize there was a character.
JSONification, that's it. That's the answer.
Can you tell I was bored when I was making these questions?
Mm-hmm. Yeah. So this is signal crosstalk. So the difference is Signal crosstalk is where different signaling pathways, so they basically, you have multiple different pathways and they work to either increase or suppress the other. That's the difference when we're talking about pathways, like you're increasing or suppressing other molecules, other cellular processes, whereas signal amplification is you're just amplifying a certain like message being sent. Like the COVID example or the gossiping example, like it gets spread around to like a lot of different, and you get like a lot of different molecules end up with that signal. Signal crosstalk is it involves this increasing and suppressing other pathways. Kind of like, yeah, go ahead.
A cell wouldn't want to like undergo like division and like cell death at the same time, for example. So, or not usually. So like the cell death pathway would be suppressed while the cell division pathway would be like increased or excited or whatever. So like you don't want like different things happening at the same time. So it's kind of like a regulatory mechanism where nearby cells can kind of like, or nearby pathways could also like influence each other.
Okay, I have one question about this. It was honestly just related to the quiz that had to do with this chapter. So I forget, there was like a certain protein that was like inhibiting something. I don't really know what it was, but basically the question was which like would occur faster, and I put signal amplification because I thought like with the gossiping it would occur like very quickly, but the right answer was signal crosstalk because they were closer together. I guess I'm just kind of confused on like how you could tell like when I don't know.
That is a little tricky. I think the reason signal amplification is a little slower because it's like it's not like a linear thing, it's an exponential thing. So like it starts slow and then it gets really quickly, whereas signal crosstalk, it could happen like in one step, for example. So like it all takes is like one inhibitory molecule to inhibit like a neighboring pathway, whereas signal amplification, it takes like a little while for that to like build up, you know. I think that's what she was looking for there, but that is a little tricky.
Okay, thank you for that. Also, I have another question. Is the signal, I know signal crosstalk is with two different signaling pathways, and signal amplification is with one, correct? That's why it takes longer because they're trying to find other like signals that are similar.
Yeah, so signal amplification is within one pathway usually, and signal crosstalk is between a couple different pathways.
Okay, thank you.
Anyone else have any questions about this?
I have a question. Did you say signal amplification is faster or slower?
It's slower, I would say. It takes longer to build up.
Okay, thank you, I appreciate it.
Any other questions? If people are still confused with this, we can come back to it after the Kahoot too, if anybody has more questions. All right.
All right, true or false? Growth and shrinkage happens at the minus end of microtubules in vivo.
Yeah, so the key part of this is in vivo. So does anyone want to explain this one real quick? So remember, in vivo means it comes from like the Latin word vivir or Spanish. In vivo means in life. So in a living thing. So which is different than in vitro, which means in like a plate or a petri dish or something. So in vivo, there's something different about how microtubules grow. They start from what's called like a structure called a centromere, and then they grow from there. The minus end is kind of like dug into the centromere, so nothing grows or shrinks like from the minus end. Everything happens at the plus end in vivo. In vitro, on the other hand, you could theoretically have growth and shrinkage at both ends, but it's faster at the plus end. Does that make sense? I can go over that again if anyone has any questions on that.
Yeah, do you mind going over that again? Sorry, just like the in vitro versus like in vivo. I'm a little.
Yeah, Rachel, you have a good way to remember that, right?
Yeah, so the way one of my teachers when I was studying for the MCAT taught me this, it was in vitro. Think of it, vitro has a T and so does a petri dish. So in vitro occurs in a petri dish. Think of that with that T. They both have that T. And then if you can remember in vitro occurs in the petri dish, then you'll know that in vivo occurs in like a living cell or organism.
And so the difference between those two things, so in vivo, so like in a living thing, microtubular growth starts from the centromere, remember? And so the minus end is kind of dug into the centromere in vivo, and nothing happens from that. Everything happens at the plus end. Whereas in vitro, everything's kind of just like floating around in the petri dish. And so there's not, the microtubules aren't like stuck into a centromere or anything. So you can have growth at both the minus and the plus end in vitro. And within that, the plus end grows much faster, but you can still have the minus end, growth at the minus end. Yep, no problem. Any other questions about that? Cool.
Which of the following is a key factor of in vitro assays?
This is a key thing to remember when you're doing the exam too, in the graph question.
Yes, definitely. Nice job, guys.
Yeah, good job. So the key factor of in vitro assays is because it's not a living thing, there's only a limited supply of tubulin and the limited supply of GTP. So like once the tubulin is like all used up, or more likely once all the GTP available in the petri dish is used up, the microtubule is just gonna like completely like depolymerize because it needs GTP to keep growing as part of that stability. Any questions about that? And like I was mentioning, in vitro, addition and shrinkage happens at both ends, and there is a lag phase in vitro because there's not an organizing center at the center. I think it's a centrosome, not centromere, as I was saying earlier. Centrosome. There is a lag phase because the tubulin has to kind of like gather together. It's like floating around, so it needs to like it needs some time to like start polymerizing, and which causes the lag phase. And yeah, any questions?
All right. Jason's the master at microtubules. That's why you always grade the microtubule questions. All right. We use spectrophotometry in the in vitro microtubule assay to measure what? I can never say that. Sorry. How awesome Jason and Rachel are is the right answer.
It's definitely
Yeah, good job. Wow, no one picked yellow. Okay. Fine. No worries.
Does anyone want to explain this one? What is spectrophotometry? You might have done this in 153 lab if you actually had it in person.
Ugh.
Sorry.
Sorry. I just hated 153.
Hey, at least we got it in person. We can't complain.
That's true.
Anyone
Have to do with absorbance and like wavelength, things like that.
Yeah. So why would, or anyone else can answer this? Why would or when is the absorbance going to be higher or lower?
When like there's a longer microtubule that's formed.
Not necessarily longer microtubule, because remember, there's lots of microtubules growing at the same time. So we're really measuring the overall amount of polymerization that has occurred within all of the microtubules. So it's sort of related to length, but more generally, it's just the amount of polymerization that has happened. That's kind of a key point that she might ask about.
Anyone have any questions on this one?
Wait, so just to clarify, like she would ask something about why we use spectrometry. Okay, I can't say it. Like
spectrophotometry, yeah.
Okay, so it's important because if the microtubule length, I thought it was like if there's more length occurring, then the absorbance rate is higher.
Yeah.
Is that what we're supposed to know, or lower, or
sort of. It's like connected. So like imagine like you have like a petri dish full of microtubules, right? And they're all growing at one time, and they're all gonna have different lengths because nothing's happening at exactly the same rate. So we can't measure like the length of like any one microtubule, but we can measure like the overall amount of polymerization that has occurred, because the more the microtubules have polymerized, the more absorbance is going to the spectrophotometry is going to have a higher absorbance, right? Because the light is getting absorbed by the more microtubules that there are. So like it's kind of related to length, because I guess the longer that they are, the more it'll absorb, but it's more related but what it actually measures is the overall amount of polymerization that has occurred throughout like the whole assay.
Okay, thank you.
Any other questions? Eukaryotic chromosomes generally only have one of which of the following? Yeah, so eukaryotic chromosomes only have one centromere. The centromere is the part of the chromosome where the microtubules attach when the spindle is formed. So they only have one on the eukaryotic chromosomes. Origins of replication, there's actually many throughout the eukaryotic DNA. They have like multiple origins of replication. So that is why that is not the correct answer. But they do only have one centromere per chromosome. Does anyone like have any questions? Want to explain why they chose what they chose, or?
Okay. The kinetic core is part of the centromere, yes?
Yes. It's like where the so the microtubule attaches the centromere like via the kinetic core. If that makes sense, like during spindle assembly. It's its own like entity, like it's its own molecule, but it is like associated with the centromere. Yeah.
Okay, thank you.
Yeah. Anyone have any other questions? Okay. This is an MCAT question, so, but it is something that you should know for maybe like this exam if she asks something about splitting, but yeah, centromere is the only one. What happens if mitosis begins in S phase? So say there's like a mutation and for some reason, mitosis begins in S phase instead of waiting until M phase. Yeah, good job. Yeah, so the genome will become fragmented and incomplete because, so what cellular function is occurring during S phase that's important to be completed?
Well, DNA replication happens in S phase.
Yep. Yeah, exactly. So if mitosis began in S phase and the cell wasn't done replicating DNA, then the cell would separate with incompleted like DNA that was replicated. It'd be fragmented. You probably end up with a non-functioning cell or some type of mutation. So the DNA repair would not fill the missing gaps because DNA repair occurs in S phase, and if the DNA replication doesn't, or in G2, like that weird checkpoint where that DNA repair occurs, but mitosis would like the cell would split before we get to that checkpoint where DNA repair would fill the missing gaps, or so it wouldn't be able to do that. The cells would not grow more quickly because it probably wouldn't produce a viable cell. Yes, I guess they would divide quickly, or like more quickly, because they aren't like they're dividing when they shouldn't be. They're dividing before like they go through all the checkpoints, but the cells wouldn't be able to grow more quickly because it wouldn't be a viable cell. And it's kind of the same way as unregulated cancerous growth. So unregulated cancerous growth occurs when the cell goes through every phase of the cell cycle. It's just mutated to go through it and kind of bypass all the checkpoints. But it wouldn't display unregulated cancerous growth because in S phase, we're still replicating our DNA. So it's just too early in the cell cycle for that to occur. So yeah, the correct answer is the genome would be fragmented and incomplete. Does anyone have any questions? During which of the following portions of the cell cycle are mRNA and proteins mainly produced? This one's also an MCAT question, so don't be worried if you don't know it, but Laura wanted me to make you guys sweat a little bit, so. This one, I think the answer is just good to know, just for your, like, may help you on an answer on the test, but it's not like critical if you don't know it, so don't worry. Yeah, so G1, so when you see mRNA and proteins, that's how you know a cell is growing. So G1 is when we're prepping for DNA replication. So when we need DNA replication, we go through the process of getting everything ready. In G1, the cell needs to grow. It needs to make sure we have all the correct molecules for DNA replication to occur in S phase. So when you see that mRNA and proteins being produced, just think G1 phase. G2 phase is a good answer too, because I could see why you'd pick that because you're still growing, but this is specifically in preparation for replication. When you see mRNA and proteins being produced, I can probably guarantee you're not going to see a specific question like this on the test. It just may be some extra knowledge that may help you answer some questions. So don't worry if you didn't get this one right. But that mRNA and protein production really occurs during G1 phase. Anyone have any questions on this one? All right. During the eukaryotic cell cycle, what is the purpose of the G2M checkpoint? Yeah. So when we're going from G to M, this is when the cell is growing. So this is where that the DNA has, where we check to make sure the DNA hasn't been damaged during S phase. We don't check that during S phase. I don't really know why. It would seem to make sense to check for it before we enter G2, but G2 is really where the repair occurs. So this is why we check that before we go into M phase. Yes, we are making sure the cell is ready to divide, but that is kind of more so like, that's a little more arbitrary, and it's more so the fact like the cell has already gone through like the like point of no return. So the cell like is going to divide regardless. But this checkpoint's really to make sure that the DNA hasn't been damaged. And the formation of the spindle fibers occurs during M phase. So like when we're doing that spindle assembly checkpoint, that's in M phase already. So it's not the G2M. And the chromosomes are aligned during metaphase of mitosis. So that's also during M phase. But does anyone have any questions on this one? It's kind of tricky. Which of the following choices will be affected by a cell containing a non-functional copy of the protein P53? P53 is a tumor suppressor, or sorry, proto-oncogene, not tumor suppressor. Yeah. So P53 is kind of the like genomic regulator of the cell during the cell cycle. It's one of the most common mutated proteins in cancer. So if we have a non-functional protein of P53, apoptotic pathways will be affected, DNA repair pathways will be affected, and the ability to arrest the cell cycle will be affected. So it is all the above. P53 does a lot in stopping the cell from going on if it's mutated. So that would make it a tumor suppressor. Yeah, it's a tumor suppressor. So it does a lot of things to stop the cell cycle from going on to a cancerous phenotype. Yeah, so these are all the pathways that be affected by it. She emphasized P53 a lot when I was in the class. Don't think she does so as much anymore. But this is just to know, like not specifically P53, but just know that there are proteins involved in the cell cycle that lead to, like, if they have a loss of function, it could lead to cancer. If they have a gain of function, it could lead to cancer. Depends on what its job is, but just know that there's a lot of these regulatory proteins involved to prevent cancer from happening. So say you have a really overactive cell and it wants to divide, but it's like has damaged DNA. The cell is a normal functioning cell, the cell will go through apoptosis and programmed cell death, because we don't want a mutated cell to continue dividing. But in a cancerous cell, if you have these non-functional proteins like P53, it won't get that signal to go through programmed cell death. So it will continue dividing with this mutated protein, mutated DNA, and possibly lead to cancer. Same thing with DNA repair pathways. If there isn't something regulating it, like P53, then it's the same thing. There's nothing to repair the mutated DNA, and then you get these cancerous cells. And then the same thing, the breaks in the cell cycle are just as important as the gas pedals. We want to be able to stop the cell cycle if there's a problem. We don't want dividing cells that are mutated, because then we get diseases like cancer. So that's the main thing with this question. It's not specifically P53, but just know that there are proteins and like tumor suppressors, oncogenes, things that regulate the cell cycle to prevent cancer. Anyone have any questions? Which of the following might cause cell cycle growth arrest? Yeah, all the above. Good job. DNA damage could cause growth arrest. Lack of appropriate growth factors is kind of self-explanatory. If we don't have growth factors, the cell's not going to grow. And then the same thing with the failure of chromosomes to line up on the equatorial plates. And it's just referring to the part like of the cell cycle where that occurs, but all of these could cause cell cycle growth arrest. Good job. What is an example of a molecule that can stop the progression of the cell cycle? I guess I did that. I mentioned that P53 can stop the cell cycle. But there's a better answer, and the better answer is cyclin-dependent kinase inhibitor, because the P53 is, it's more so in terms of if there's a problem, like it's a tumor suppressor, so it suppresses tumors. But cyclin-dependent kinase inhibitors are always active, like if you need a break, like in the cell cycle, because if you remember, CDKs are like the main regulators of the cell cycle. So if we need to stop the progression of the cell cycle, cyclin-dependent kinase inhibitors are going to be the most effective in doing that. So that's why that's a better answer than P53, because P53 isn't, like, in the sake of this class, you really don't need to worry about it. So I guess that last question was a little misleading, because it definitely does stop the cell cycle, but CDK inhibitors are the better answer because they're more likely to stop it, because they're always available, really. P53, no, go ahead.
They're used more often, I guess. P53 is, I think of it more as like an emergency stop.
Yeah.
CDKIs are more likely to just like be regulated like more often.
Yeah. Yeah, exactly. So that is why CDKIs are the correct answer. Do you guys have any questions? I definitely threw you off with that last question. I do apologize, but that's a good way to think of it. The P53 is the emergency brake. Cyclin-dependent kinase inhibitors are just the brakes. Does that make sense to people? Okay. I think the problem with this question was I had to shorten it. So I think it had like a scenario, which is why CDKIs were the correct, like better answer, but I had to shorten the question. But okay. Neural cells cannot regenerate. During which phase of the cell cycle are neural cells arrested in? Yeah. So G0 is that phase where we have non-dividing cells just kind of in, because so neurons don't regenerate really. That's why when you get like dementia or something like that, or a spinal injury, it's difficult to treat because they don't regenerate like other cells do, like our skin cells. But neural cells, they are always arrested in G0 because they don't need to go through the cell cycle, which is why they're not in G1, because G1 is when we have a cell that is getting ready to divide. Neural cells never even start that, so they're always arrested in G0. But most of you guys got that right, so that's good. All right, last question. Colchicine is an anti-inflammatory drug. It is able to do this by... The correct answer is green, if anyone's wondering.
I'm going to put the final question in the chat. This is the most important question.
Yeah, I can't believe I forgot it. I'm so sorry. So colchicine is one of those drugs we were talking about before that it's a good idea to know, because she may ask you to draw that diagram with it. It could be Taxol or vinblastine too, but this is one of the three. Good job. Good job, guys. All right, yeah, colchicine inhibits microtubules. That's how it stops inflammation. Fun fact, I always tell everyone this, Jason knows this, but I do take colchicine, so I am obsessed with learning about it because I'm like, what is it doing to my body? It's an anti-inflammatory drug. I have an autoinflammatory disease, so it basically keeps me alive by doing this. It the thing with inflammation is it causes like this whole cytokine storm and this whole thing, and it's really complicated. But basically, colchicine inhibits microtubules from forming or from like being that dynamic instability. It prevents that because it prevents the cells from pretty much dividing, because microtubules can't attach to the centromeres and we can't have cell division. So it kind of stops those cytokines from forming or generating when it's not needed. But yeah, so the main thing to know with colchicine is that it inhibits microtubules. It does also kick Jason in the butt, but you know. All right.
So just to clarify, for like a an SI like graph or whatnot,
Yeah.
If it were to be like with colchicine, there would just be like a straight horizontal line because there can't be any growth if they can't attach to like the centromeres for the origins of replication, right?
Jason, that is you.
So yeah, pretty much. It won't be able to continue to grow because it just inhibits microtubule growth. So it'll be more of a straight line. It might like grow like a little bit because colchicine, it won't be like entirely effective, but it'll be pretty much a horizontal line.
Okay, thank you.
So that was it for the Kahoot. Does anyone have any specific questions? We also have like Professor Francis gave us some like open response questions we could ask, but it's up to you guys. And if not, thank you for coming. Like, we hope you guys do well in the exam. Please do open response questions. Awesome. Okay, sure. So, all right. So the first one is,
I'll like paste them in the chat.
yeah. So Jason's gonna put it in, but I'll just read it. You have found that RAS has a gain of function mutation and you decide to treat the cells with an RTK inhibitor. Will this help? Yes or no?
And also, we don't have the answers to these, so we might have to like work through it with you.
Yeah, we do.
That was me, so they could be wrong.
Oh.
So,
well, no, well, that's right. The first one's right.
Yeah.
No, yeah. Yeah. No, it won't help. Does anyone want to, you can put it in the chat or you can say, but like, why do you think it won't help?
Is it because the RAS activated, the gain of function of the RAS will only affect downstream protein, like it will affect the MAPK3, MAPK2, MAPK1, but it wouldn't affect all the upstream protein, like GRAP2 and the RTK.
Right, exactly. Yeah, that's kind of like what I was talking about earlier, where like if you have something that's already inhibited upstream, like if you add something downstream, it might not have an effect of whatever you want trying to do.
Yeah, so the main thing, like if RAS is already downstream of RTK and it has a gain of function mutation, that means regardless of the signal it receives from RTK, it's going to be active. So inhibiting RTK won't affect RAS if it has a gain of function mutation because it's already downstream of it. So it's like if RAS is constantly on and you send a signal through RTK for it to stop, it's not going to do anything because it's going to be active regardless. So yeah, the correct answer is no.
Cool. All right, next one.
Yeah. If you have a gain of function in GEF, will the signaling pathway be more active in cells with wild type GEF or mutant GEF? So that just means, will this pathway be more active in cells that have a normal functioning GEF or with this gain of function GEF? Mutant. Yeah, so when we worked this out, we also came to the conclusion that it'd be mutant. Does anyone want to say why they think it would be mutant? You can either put it in the chat or say it, or we can go over it. It doesn't matter.
doesn't GEF change the affinity for GDP, so it makes GDP diffuse out and GDP diffuse in. So if GEF is more active, then there's going to be a more active pathway because there are more activated proteins.
Yeah.
Exactly.
Yeah. That's basically it, yeah. All right. So the next one, does the receptor in the JAK-STAT pathway have kinase activity? No, we got one no. What do you guys think? Three no's. All right. So
I'm not completely. I think it's yes, because
yeah,
I think the JAKs kind of phosphorylate each other, and they kind of are the receptors, and then once they receive a signal, they phosphorylate each other, so they do have kinase activity.
They do, yeah.
Sorry, I have a question.
I thought she went over an example in class because we had to do a question, her little questions that she had from each week, and I remember this one being no, because she showed the image where the receptor, it was the two JAKs on there that had the kinase activity, but the receptor itself didn't.
Okay, that could be true.
Oh yeah, no, you're right. The receptor itself is different from
Okay, the JAKs aren't the receptor.
The JAKs aren't the receptor. No, you're right. You're right.
Got it. Yeah, these are the questions you went over in class, so that's probably right.
Because the JAKs, yeah.
So yeah, I guess that makes sense. So the receptor will receive the signal, and then that will tell the JAKs to then phosphorylate each other. So the receptor itself doesn't have the kinase activity, but the JAKs do.
The JAKs do.
Okay. Thank you.
Because the name of JAKs are Janus kinases. That's what they're called. So I think that's where we were thinking with that. But the receptor does not. The JAKs do. So yeah, the correct answer is no. Sorry about that, guys, but good catch.
I have a note from another review session, and it says that they're not considered to be they don't have a kinase domain, but they're receptors. So
yes. Okay.
Yeah, I mean the JAKs themselves do, but the receptors that trigger them do not.
Yeah, because JAKs are
kinases themselves. That's their name.
They're not. But they're called non-receptor tyrosine kinases.
Yes. Yeah.
Okay. Yeah. Which is different from an RTK, which would be a receptor tyrosine kinase. Okay. Yeah. That makes sense. Sorry for confusing you guys. Thank you for the correction. All right.
All right. Next one. Would a loss of function mutation in P10 lead to cancer? You guys remember what P10 is? So P10 is involved in the AKT survival pathway, right? It's involved with that PIP3, the lipid that's in the membrane. So P10 itself is a tumor suppressor. So when it's functioning, it's preventing cancer. So if we have a loss of function of P10, that means that it will lead to cancer because we don't have that thing, we don't have that line of defense of P10. Because its role in the AKT pathway is that I'm trying to think of how to explain this. So if a normal P10 is involved with like phosphorylating PIP2 to PIP3, right? And then PIP3 can go on to PDK1 and AKT and lead to whether cell cycle or cell survival or whatever it needs to do. If we lose a function of P10, PIP3 won't be phosphorylated, and we don't get that possible, like, what's it called? Regulation with apoptosis within that pathway. It's kind of hard to explain, but P10 is a tumor suppressor. So if you lose a function of it, you're going to get cancer. It's actually one of the most common mutations, P10, loss of function in like breast cancer, lung cancer, all that stuff. Does anyone want to like explain why they think what they think? Or I know that wasn't a really good example. I'm just trying to think of how to explain P10 without getting into like cancer pathways, because there's a lot of them, but it's involved in that like AKT pathway, the survival pathway. Does anyone?
Yeah.
Let me try. So P10 converts BIP3 to BIP2, right? Then
I think it goes back and forth between PIP2 and PIP3. So it could cause either. Like it can, yeah.
Oh, I thought you said like on Monday or something about PIP3 to PIP2, but
it might be so we talked about this. So I'm in a cancer genetics class, so that's kind of why I'm like thinking of it deeply, but Laura might have mentioned that it goes from PIP3 to PIP2. I've learned that it goes to both. But yeah, this one's kind of tricky. She sent us this one, right, Jason?
Yeah, this is one of the ones she went over in class, so she might have a better explanation in one of the videos.
Yeah.
She just sent the questions, no answers. So
we have a question. Can we go over regulatory proteins that can end up causing cancers that she might ask in scenarios? Yeah, we can do that. Let me actually pull up the exam from last year or last semester.
Well, the next question is kind of like that. Hold on. So the question is, I'll post it in the chat. If P10 has a loss of function and cells are surviving when they shouldn't be, would it help to treat them with a bad inhibitor? So,
that's the type of question she would ask.
Yeah. So like think about 100%. What bad does. See a couple of yeses. So remember, as we were talking about earlier, BAD's job is to inhibit BCL2, and BCL2's job is to inhibit programmed cell death. So if you inhibit BAD, then you won't have the sequestering of BCL2, and you would stop it with, so if you inhibit BAD, it will not sequester BCL2, which would allow BCL2 to plug up the mitochondria, which would not cause cell death, right? So that wouldn't work if they're surviving when they shouldn't be. I think. So like BAD's job is to, it kind of like, so BCL2 stops cell death from happening, and BAD's job BAD is bad, so it inhibits BCL2 from stopping cell death, so it would allow the cell to keep living. These double negatives are tricky.
Yeah. An example of like a question with like molecules or regulatory proteins that can end up causing cancer. So one is RB, so that like blastine protein. So that's like a known tumor suppressor in humans, right? She like mentioned that in class. So in many cancers, the RB gene is observed. So it's been observed that both copies of the gene are mutated to a loss of function. So that's the main thing. When you have a loss of function in tumor suppressors, it leads to cancer. If you have a gain of function in oncogenes, it also leads to cancer. So it's kind of like if you lose that tumor suppressor, you're losing that line of defense. And if you get a gain of function in an oncogene, you're like adding more to like adding salt to the wound, kind of, right? So if we express the RB gene at high levels in all human cells, which is a tumor suppressor, right? Would this protect humans from cancer? So this is like a question she's asked on previous exams about this RB gene. So this is kind of like one of those examples of a type of molecule or protein that causes cancer in humans that you kind of would need to know like the pathway that it's involved in to answer the question. So like what molecule is like RB involved with? Like what is one of the molecules we talked about that it's like works with?
Is it EF2?
Yeah, E2F. Yeah. Yeah. No, close enough. Yeah, so that's the first thing you would want to think about when you try to answer this question. So you would need to know like this pathway, like how their restriction points work. That's just kind of like a type of question that she could ask involving like certain molecules in cancer. She doesn't spend too much time on cancer, to be honest. The cell cycle is easy to ask about like with cancer questions because that's how cancer usually forms, is from like problems with the cell cycle. But that's kind of like one of them that she could ask about, like RB. I doubt she'll ask about P53 because she didn't really spend much time on it this year. But that's like one of them, if that answers your question.
I would just kind of like go through the pathways and make sure you know what's happening normally. And then try to just like play around with the pathways. Like if something is overactive, what would happen? Underactive, what do you think would happen? That's the kind of question that she's gonna ask. I can't just, unless you have like a specific example, I can't just like go through it, but
there's actually a website. I'm trying to find it. There's a website. It's like used with like cancer therapies, but it's like it has a bunch of pathways and you can like go through and like try knocking some out, like certain molecules out, and it'll tell you like what will happen. I'm trying to find the link for it because we used it in the cancer class, but when I find it, I will definitely send it to you guys because it's actually really helpful to just go through and like you can just like try knocking out like different pathways or different like molecules in the pathways, and like it kind of just shows you like how it works like without that molecule or if it doesn't work. I'm trying to find it, but
yeah, that's one of the ways that online classes will be a little easier because I would have like the diagrams like already when you're taking the exam, because you can like look through the diagram, see what's happening at each step. I would also kind of like write out the steps of the pathway so you can like visually see what's happening if at each step, and like what would happen if that step was messed up and like the order and stuff. I know Rachel used to study for the exams by like drawing out all the pathways.
Yeah,
that could be helpful.
I like to draw them out just because I'm like a visual learner, but yeah, whatever like works for you. Definitely try it. The main thing with exam 2 is like pathway. What happens if you knock this out? What happens if you increase the function of this one? Like all this, like you got to know like what would happen to the pathways if they were dysfunctional and what would happen if they're like overactive or something like that. So that's like the main advice we can give for this one, for this exam.
Yeah. So anyone have any specific questions Renarto can stick around for like 5 or 10 more minutes.
Yeah.
I have a very, very stupid question. What is the difference between pre-RC and pre-IC? I thought they were the same thing. Or are they the same thing?
No, they are not the same thing. So the pre-RC is the pre-replication complex. This is like the start of the DNA replication in like in S phase, like our early S phase. The pre-IC is the pre-initiation complex. There is a difference. The pre-IC, so the pre-RC is like you're preparing for DNA replication. The pre-IC is that one's like more difficult to explain, but they are different. The pre-initiation complex is kind of like transcription factors, if that's a good way to describe it. I don't know, Jason, do you have a better example?
I don't. This is not my strongest area.
Yeah, Sam.
I think I got it. I got it, to be honest.
Okay.
They are different, yeah, but
yeah, I just got confused between the two of them. I thought they were the same. But
yeah. Sorry, we can't explain better. That one's also not my strongest suit, but I'm gonna try to find that website. Yeah, of course. And see if I can, then I'd send out an email to like the whole class with it. But it's like a pretty helpful website where you can like knock out certain molecules in a pathway and see what happens. But yeah. Thank you guys for coming. Good luck on the exam.
Thank you. Have a good evening.
You too.
Thank you.
Do we have to do check-ins for this? Like,
yes. Yep.
Yeah.
The only thing this week is the exam and check-ins, I think.
Yeah. Okay. There's no discussion. Yeah.
Okay. Thank you, guys.
All right.
Bye. Have a good night.
Bye.
I'm so tired.
Oops. You want to stop the recording?